Okay, well, thanks for people attending another week of our meetings. Next week I'll have to cancel. I'll be on a trip, but just to give you a heads up, I'll send a notification and calendar cancellation for next week. But then we'll resume the following week. Yep, have the in-person meetings always. People have any ideas to try to have an in-person meeting for our group, and I'm always open to that. No new publications as I'm aware from our group yet, but I know there's some under review or like preprints here. And again, just a quick update as I bring up every week. There's the case reports that we're working with Al. He has some interesting case reports with long COVID that if you're interested in helping us out, there's lots of data to go through from these patients that suffering some unique long COVID conditions that will be helpful if you want to join. A lot of data to look at it and try to figure out what's happening. And it'd be a nice case report to write. And of course, everyone would be an author on the paper who helps. Okay, let's dive into the news. Last week, we briefly talked about the XBB, the two new variants that have been popular. One of them is that XBB.1.16 that's now responsible for this spike in India. The other one's XBB.1.19. Has it 9.1? I think it's 9.1. That one's in Singapore that you can see the spike. But this XBB.1.16 we talked about last week briefly. They're calling it Arcturus. In India, that's what they're seeing, the big spike. So this is an article just going over it again, talking about that there's, as we talked about last week, due to a couple of the mutations that have occurred from the evolution from the XBB.1.5, that people are thinking that it's going to be 1.2 times more infectious from the last one, which is the XBB.1.5, which is causing this huge spike again. The interesting thing is that people don't see for adults not too many differences in symptoms, that we see with the current variants that are, or the current dominant variant, the XBB.1.5 that's been circulating. But this article here is the first time I heard about it, which is pretty interesting, is that this one Indian clinician is starting to see these unique symptoms happening in infants that previously have not been seen before with SARS-CoV-2 infection, which they're saying an infantile phenotype, he's claiming, and then he's saying basically they're saying things like conjunctivitis or pink eye or high fever, cough, and itchiness in infants. Adults, for some reason, don't have this, so they don't know why. This is just an observation that they're seeing happening in India. So I guess we'll see if this keeps happening, and maybe there might be some papers in the months to come to figure this out. Why this variant is causing this and the other ones didn't, I don't know. So go ahead, Kevin. Yeah.
Yeah. Did they report hospitalization rates across lifespan?
In India, they're saying the hospitalizations from the articles I've read. I mean, the numbers are still kind of rough, but it's about the same as the previous variant for adults.
Okay, thank you.
Yeah, mainly because they're saying the symptoms in adults seem to be the same as the previous variant, and the immune evasion is not any different from the previous variant. What is known now? So I don't know if that will change.
There's no increased hospitalization for pediatric cases.
Well, this is the case. It's saying for the infantile phenotype, they're saying they're not necessarily hospitalized, but they're saying the conjunctivitis and different kind of high fever and cough and itchiness. So whether they're hospitalized because of this or not, I don't think so. But that's what the symptoms is just that. Yeah, for the infants.
Okay. All right. Thank you.
Yeah. No worries. So I guess more to come to see how this might develop. Something that's relevant here is that, Biden signed the ending the COVID national emergency that we know is coming. So the impacts of that we'll see in the months to come. But this also impacts, this should we get, and I've discussed this last week and the previous week, should people get another booster shot, as most, I think, scientists that study this agree that every 6 months would probably be a good idea to get your booster because we know of the fading immunity for the bivalent and other boosters. And the Scientific American article just talks about, that and talks about the pros and cons of it, and then the lack of guidance from a lot of places, especially in the U.S. In the U.S. mainly, other than last week, the FDA said, yes, if you're immunocompromised, older, and if you're in the high risk category, you can be allowed to get the second bivalent booster. Places like UK and Canada are offering it to, I think, everyone in the high risk category, older population. But the WHO recommends, additional booster of 12 months or 6 months for that. So the point of this article is there's not much guidance from the government agencies, but a lot of scientists are saying, you probably should get a bivalent every 6 months for now until other additional vaccines or things come up to tell us otherwise. So you can read that. Talking about vaccines, this paper came out a few days ago, 3 days ago. This was an interesting, I didn't know whether this was in circulation or they're working on this, but they're working on a T-cell vaccine for COVID-19, this paper here. It's a couple of figures. It's a short paper. And in this paper, they basically say, what they've done is making a, as opposed to the standard vaccines, this one here as targeting predicted to be like the MHC class and T-cell response, and using that as they call it RAVEN, rapidly adaptive viral response, antigen presenting cell targeting. So that's basically, this is a figure from the paper that they basically are showing. I'm going to show the two figures from the paper showing the responses. So this paper here shows that, using this newly designed vaccine in mice, so they've only tested it in mice so far. They see, it induces a T-cell response, as these schematic and figures show here, and the several hotspots related with the the histocompatibility complexes are being where the predictor places that this vaccine is binding to. Go to the next figure, and then they tested in the C57 background mice, and then the background mice that are we all know that are used for SARS-CoV-2 research. And what they see here from the mice studies, that this is the response to the after infection, the SARS-CoV-2 with and without the vaccine, the survival rate is much better. I mean, it's remarkably better, as you see in panel A. And then this shows basically the pathology of the lungs and how with and without the vaccine, the improvement you see. So this has seemed to be an interesting article talking, promising potential newer vaccines, because one of the things in the Scientific American article talking about, the need for probably better vaccines out there that would be more targeting the direct places where the virus come in, or better responsiveness and last longer. Last week we also talked about the nasal vaccine that's being developed that has seemed promising. Some long COVID news. This is mitochondrial relevant. I just saw it. This paper just came out just today, so you probably haven't seen it until now.
I'm a little behind.
Yeah, so it came out I think this morning, so you might have not been paying attention to it unless you start scouring the internet like I did. So this is interesting. So I thought I'd bring it up now. I imagine Doug, as you, I don't know if you know about this drug. I've never heard about this drug, but you might have. But what they've done here is they tested this drug that's related with, I guess, targeting the metabolism and energy production of cells, or increasing it, I should say, and then reducing inflammation. So they're using it. They're saying, since long COVID induces fatigue and everything, and according to what Doug theorizes and we theorize, that long COVID is probably mitochondrial related disease. It looks like these researchers believe the same thing. And these guys are from the University of Oxford. I don't know any of them, but I'm sure some of you might. Basically they're saying they didn't do any molecular aspects, at least in this paper. These are the two figures from the paper. They didn't look at any biology, but what their assessment of what this in the drug, when they applied it to the patients, they're looking at basically things like mental fatigue, physical fatigue. They looked at physical characteristics like walking and things like that. And it's a small study too, so keep that in mind. That was, they say, 41 patients twice a day for 4 weeks. So that's a small study, and they say that in this paper. It's saying it's a small study, and we need to probably the study. This showed promising results, basically in improving mitochondrial functions and reducing the impact that long COVID supposedly impacts mitochondria. Again, they didn't show any molecular things about mitochondria, but they're claiming that's the case based on some references they had. And So the improvement they see here. So I don't know what people know about this drug, the AXA 1125, which I do not know anything about. Maybe you want to comment more or not.
I'm trying to get some.
Oh, sorry, Doug, go ahead. You got cut off.
I just said I'm looking it up right now, so go ahead.
Oh, okay. Okay, yeah. Sounds good. But I thought this might be of interest. So again, no biology in this paper, just purely they gave the drug to patients and they saw the outcome without looking at any biology. But looking at how they've improved in the clinic. So, but it's promising. They say they saw statistically significant improvement even in this small cohort. So this theory that it is a mitochondrial disease, long COVID, these guys are supporting that hypothesis. Some other long COVID news. This is interesting because I guess the question here was at least I would have thought that the more times you get COVID, potentially the more chances you might be increasing your risk of getting long COVID. That's some people think that, some people didn't, but it was unsure. So this is a UK report from one of the government agencies there that they looked at a large cohort of long COVID patients who were suffering long COVID, and again, they gave basically questionnaires out and looked at EHR records. So this is purely epidemiological type study. So what they did, and it's a large cohort of people that got to answer these questionnaires. And what they saw is that looking at this large population of this is the dates in the 4 weeks in March. So this is the very, very recent from February to March. In the most recent, cases of people with long COVID from February to March, if they got infected more than once, it actually shows that you might have a 2.4 chance. In this survey, they say there's a 4% chance for long COVID symptoms persisting at least 4 weeks, after your first infection, which is a lot lower than some previous numbers that we know. But this is most recent, so these people are vaccinated, and this is the most recent variants might be related, Omicron variant. And then they said people who were infected a second time, actually the chances were 2.4%. But there's some things that you have to say in the study where they didn't address, possibilities. The study excluded that, people have got long COVID for the first infection, so didn't get it. The first infection may be less prone. They didn't address that. And the study also says didn't examine the second infection worsens symptoms in people. So these things weren't covered.
Afshin, may I interrupt?
Yeah, go ahead, Judge. Yeah, please.
So AXA1125 is a mixture of 5 amino acids: arginine, glutamine, isoleucine, leucine, and valine, plus N-acetylcysteine. And some of our colleagues, remember, more than 2 years ago, suggested N-acetylcysteine might be interesting. It generates glutathione. So this is really N-acetylcysteine is for redox, glutathione. Isoleucine, valine, and leucine are branched chain amino acids from the branched chain keto acid dehydrogenase. So they will feed carbon into the TCA cycle. Arginine and glutamine. Glutamine would get deaminated to glutamate, and that gives alpha ketoglutarate that goes into the TCA cycle. Arginine's important, could be important in generating nitric oxide. So that's what that is. So it's sort of all of those are probably can buy at the health food store, which is probably why they could go ahead and do a trial without any preclinical data.
Okay, I see. But you need the right balance, probably, as they probably were figured out somehow.
I imagine they have some concentration, but I doubt that's that critical. Thank you.
Right. I see. Thanks, Doug. And then I see Glenn had. Where's the chat? Glenn had put some more information on it too, with the 5 mixtures that you just mentioned, Doug, in the chat, and the link there. So thanks, Glenn, for that.
Sure. Yeah.
Okay, so I guess we'll have to wait and see what happens here for long COVID with this, but this pilot study seems promising. Good news, I guess.
About
Yeah. Go
This
ahead,
this
Hannah.
paper, I'm just a little bit. No, sorry, not that one. The following one.
This one.
Yeah. I'm just contemplating the 4 weeks. Does that mean they collected surveys over a 4 week period? Because obviously you can't have 2 COVID infections and develop long COVID within a 4 week time span.
No, yeah, they collect. Sorry, yeah, they collected the surveys over the 4 week period. Correct.
Okay, so they're just, in other words, they're assuming that people are correctly reporting their COVID history. Like they're not double checking. Oh, did they really get diagnosed with
No.
a first infection or whatever? They're just
Right.
assuming that the survey information is reliable.
Right. Yeah. So there's a little some things you might have to take a leap of faith with this. I agree.
Okay, thanks. Just curious.
Yeah. No, those are very good points, Hannah. Yeah, exactly. Yeah. So take that with a grain of salt. And I mean, I guess this is sort of, I think, coming out of the CDC version of the UK, if I'm not mistaken.
Well, I'm not saying. I mean,
yeah.
I'm not attacking the conclusion. I mean, I hope the conclusion is right. Actually,
Right.
God willing, it's correct because there's so many people who are getting reinfected. Let's hope that doesn't increase their risk of long COVID.
No, but I mean the limitations of the study. They actually talk about some of the limitations if you go in that all the way down, and they don't mention what you did, but they mention some of the stuff here. But I think one of the limitations is exactly what you said. Yeah. But I mean, it maybe warrants further investigation to prove if these numbers are actually correct or not, and maybe a more reliable, other than surveys.
Well, yeah, I was just thinking about in the N3C context, trying to tease out. I mean, even getting, even trying to be careful about what you consider reinfection. The way things get coded at a medical encounter, you might even have difficulty determining whether these are persistent symptoms from the last infection or is this a new infection that's being reported. But the way, those two things could get coded in exactly the same codes if you're unlucky. So it's really hard. It's really hard to get a reliable data set for this, whether you're doing it with surveys or whether you're doing it with medical records. I guess maybe the best thing would be a combination of both of those things might get you the best reliability on your data.
Mm-hmm. Makes sense. Oh, thanks.
And out of curiosity,
sorry,
did they assess a treatment?
No. They just purely talked about infections that they got between the first and second one, and that's it. And their symptoms in the questionnaire.
So the respondents were all unvaccinated, or
no. These are just there's a mix. I think most of them were vaccinated, from what I understood.
But they didn't assess that. They just aggregated the data.
Yeah, exactly.
Okay.
Yep.
And what was the demographic breakdown? Did they do any analysis involving that?
I didn't pay attention to that, but there probably is somewhere in there. There should be.
Okay.
It's the UK population, so that'd be that UK whatever demographic in there. Looking at around the UK. This next paper might be of interest to the inflammation group in the paper they're working on. So this is a preprint that just came out 2 days ago, which is not necessarily good news for people who have who get infected. But the other one was good news. This might not be such a good news. So whether this preprint holds after review, but the data seems pretty solid so far. But they're talking about basically they looked at basically how well they discovered that autoantibody production, and even after mild SARS-CoV-2 infection, despite your vaccination status for a patient, can be, it is impacted, basically after SARS-CoV-2 infection, and it can impact you with if you have long COVID or not. So basically what they say here, this is a little snippet, I think important point from the abstract, is there here we demonstrate that mild SARS-CoV-2 infection increases autoantibodies associated with lupus or systemic lupus, which, we've seen and other people have seen from the beginning, similarities from immune and inflammatory profiles that are oddly similar to lupus, not oddly now, but back then it was. But then they're saying, that the inflammatory myopathies in long COVID patients with persistent neurological symptoms to a greater extent than the convalescent controls at more than 8 months post-infection. They see these happen. So, in this kind of like the graphical representation of their study. So they looked at basically people who had neural PASC would be, post acute sickle like COVID patients, who had pathology type symptoms for long COVID. And then they looked at COVID convalescent controls and then unexposed healthy controls, so non-COVID infected people. And this is one of the figures from here. And basically what they see is that, again, antibody production is enhanced after mild COVID infection, despite your vaccination status. And then mild SARS-CoV infection increases antibodies associated with the systemic lupus, erythematosus, and then inflammatory myopathy. So, and then what they also, in the discussion, they talk about, I thought this was one important snippet I took out of the discussion, saying like over 94% of neuropasc and 100% of convalescent controls in our study presented with mild acute cases. So these weren't, people who were hospitalized or severely impacted. And despite of this, like 88% of the PASC and 95% of the non-long COVID folks had statistically elevated markers for multiple autoantibodies after infection that did not decrease, after vaccination either. So this could, and this was despite any kind of, pre-existing comorbidities, and the population of patients were fairly young in their 20s that they looked at. So this is an interesting preprint to take a look at.
Is the cohort large enough that this looks pretty solid?
So the cohort here is it's N equals 17, 14, and 15. So that's the other part of the study that if I was a reviewer, I'd be like, well, it's a little low. But the results are statistically significant. So I don't know what that means.
I think it means we need a larger study.
I don't know if that pleases your question here. What's that? Sorry, go ahead.
I think it means we need a larger study, but
right.
that it's worth pursuing.
I agree. Yeah. I think that's probably the correct assessment. I think their last statement in their abstract was
certainly option when you look down the list of things that they see. The most larger cohort in which we see that is the autopsy tissues and nasal swabs from the ongoing inflammation group work. I mean, a lot of them are the same genes in the double-strand RNA recognition, TNF-alpha, IL-6. So they may have some additional things that they're seeing. But, I mean, the genes they're seeing are what brings up lupus as a very high pathway and what DN does and everything. So not that. I mean, this is long COVID. That's what's different. We're looking at a more acute disease. But, yeah.
Yeah. I figured you'd mention the similarities. Yeah.
Yep.
The data set, actually, you can access the data set, but since it's a preprint, they say upon request, so they're not forced to actually make it. But I mentioned if we email the author, if people want to get a hold of this omics data set, we might be able to get our hands on it. But yeah, I thought this was an interesting preprint nonetheless. Let's see if it changes or they do anything once the paper is actually published. And one thing that's sort of related, this is the last long COVID news. In this Nature Reviews of Rheumatology article, they basically are talking about referring to other papers that have come out. The risk of basically saying, they provide a short little review about talking about how after COVID-19 infection, there seems to be a higher risk or higher occurrence of autoimmune type diseases occurring. Not talking about long COVID, but just saying, something that could potentially be related to this or potentially be related to, other factors. So this is just kind of a short little review going over the current literature on this and showing that it has a potential increased risk of new onset autoimmune diseases after you get SARS-CoV-2.
As you often do, Austin, could you put these in the chat? This would be a nice one to look at. And
Yeah. Sure thing.
Thanks.
Yeah. Let me do this really quick as we're talking. I gotta stop sharing though, but then I'll reshare it quickly. But then here, let me put this link in the chat for that last paper. All right, so let me go back to reshare.
Afshin, was the autoantibody paper, not this one, but your previous slides paper? Yes, was the treatment group you have a box, text box on the right that states the controls were individuals in their 20s without comorbidities. Were the infected group a similar demographic? I'm assuming so, but they were. Okay.
Yeah. Let me put this one in the chat too, so in case you want to see the details. But yeah, Sonny, that's correct. Yeah, they did, at least they did a pretty good job for age matched, for the controls. Yeah, because that would be an important point. If they weren't age matched, then study might need a little more insight or more experiments to look at.
But generally, healthy age group.
Yeah, exactly. That's what the point they wanted to make. It's not, these people previously were not suffering from any type of existing ailments, basically. And they're young, they're young and healthy. Yep.
Thank you.
Exactly. Worries? All right, I'm going to go over the usual case numbers and things like that, but I don't know if anyone has any other questions on any of these papers, or I put this one in the chat and this one in the chat for the links for these, in case you want to look at them before I send you the slides later. Well, feel free to interrupt if there's anything else. So the variants around the U.S. still, as this one shows, and I'll show you other ones. XBB.1.5 is the most dominant one, but you can see .16 here, and this is the one from Singapore, .1.9.1, that's causing the spike in Singapore to happen. This one's the one in India.
That's the same as in India.
So India's the XBB.1.16. This is this one. Though .1.9.1 is different, but they're very, I guess they're maybe Nidia. Is Nidia here? Do you know the differences in their mutations, how close they are?
No, I don't know the exact number of mutations that they differ from.
And do you know if one has a better growth advantage over the other? Because I don't know that either.
I haven't seen anything on that either. Yeah, let me look it up.
Yeah, thanks. I know they said for .1.16, as that previous article said, it says like a 1.2 roughly growth advantage. I forget what this is. You haven't really seen much numbers.
So it appears there are maybe 50 or more different variants, and I'm not a virologist, but for other viruses, are there that many variants?
Yeah, so there's all these other variants are there, but they're basically very low and they're not out competing what's dominant. Yeah. So there's tons of other variants out there always floating around for SARS-CoV-2, unfortunately. But as you point out, they're very low amounts in this population. I think that's everywhere in the world. Once it starts getting a little higher, like this 4 or 5% in the States, and in India, they're accounting for, it went from 11,000 cases to 40,000 cases now since a week. This is starting to outcompete this XBB.1.5 in India. But there's all. Yeah, sorry. Go ahead, Sonny.
No, my question was more specifically. So, is it unique to SARS-CoV-2 having this many number of variants, or is that typical with viruses?
I have to refer to Nidia.
Yeah. So it's the usual behavior of an RNA virus to produce lots of diversity. The thing with other viruses is that usually a couple of those lineages dominate a season, whereas with SARS-CoV-2, you see multiple lineages co-circulating. So it's not so much on the magnitude of the diversity, but how they co-circulate or dominate.
And the co-circulation parallel patterns are unique to SARS-CoV-2 then? There hasn't been a virus similar to SARS-CoV-2 in that regard?
Let me think, but not to this extent, no. Like from the main respiratory viruses, yeah, I don't see one that has so many co-circulating lineages.
Even the SARS-CoV-1 didn't have as much, right?
No, yeah, and it was also a very short-lived outbreak, so it didn't even have the opportunity to generate as much diversity as SARS-CoV-2.
Right.
But doesn't influenza have a lot of co-circulating variants?
So there are, so it generates a lot of diversity, and there are a couple, a few lineages that co-circulate, but not to this extent. Like we do have a few, exactly, seasonal influenza. There are some clades that co-circulate, and then we measure their growth for the vaccine updates, but it's not like measuring the growth of, I don't know, 20 or I don't know, maybe here it's more 100 co-circulating lineages, because there are definitely a few that dominate each season.
Mm-hmm.
Well, and don't they follow kind of predictable patterns of spread throughout the world, so they can kind of say which ones are coming,
right?
Potentially.
Yeah, that's also true. Yeah, they follow the air travel flows, and there's also been established that Southeast Asia is the epicenter for the generation of diversity, because they have flu throughout the year, and then those clades. The most fit clades are the ones that then seed the seasonal outbreaks elsewhere.
Nydia, why would that be? Like, why wouldn't there be flu throughout the year in other locations?
I think that goes more in what factors can also drive influenza dynamics, and we know that low absolute humidity drives influenza transmission, and those characteristics don't happen everywhere in the world just because of different climates. And in Southeast Asia, they have the perfect setup for continuous circulation of flu. There might be other factors. I don't know if population density or other things might be at the root of having Southeast Asia as the epicenter for flu diversity. But we see that it's different with SARS-CoV-2. We see Brazil was generated a very big variant of concern. The UK, South Africa, we don't have a specific geographical location that usually generates diversity.
So are there certain adaptations that people know about in coronaviruses that are for certain locations, and particularly in SARS-CoV-2? Like, are there certain mutations that we looked at this a little bit, Nydia, but like that occurrence over and over again in the same location, like to do with humidity or temperature or stuff like that?
Are you talking about mutations, or are you talking about factors that might be I guess driving the generation of diversity? Because in terms of factors, most people think that these variants of concern emerge when there is lots of uncontrolled transmission. So, and the second one is in immunocompromised populations. So I think those are the two.
Yeah. So you're saying the environment doesn't drive what the changes would be?
I don't think so. At least it hasn't been described. It's mostly at the epidemiological site or the host site. And this is also what, just going a bit off topic here, one of the concerns with SARS-CoV-2 spilling over to animals is that there might be uncontrolled transmission going on in animals, as we've seen for the mink farms, and that can generate also new diversity that then can spill back to humans. So yeah, uncontrolled transmission, immunocompromised status have been the ones that have been pointed out as the most important things.
I guess that's the other unique aspect, the zoonotic, right?
Mm-hmm.
Spill over both ways that other viruses like influenza don't do.
Well, influenza No, it does, but it's not studied. So
Okay.
there has definitely been lots of influenza spillovers from humans to animals. And since the 1919 Great Flu pandemic, the
Mm-hmm.
classical swine influenza
Mm-hmm.
lineage was initially spilled during that period. And then in the 2009 pandemic, again, the same spillover from humans to animals. But we are starting to see that animals, particularly livestock that are that live in a high density and are likely immunocompromised, humans that handle those animals sometimes are infected with the lineages that have been evolving in those animals. And there are a few papers describing that, but because this is all this has to do with the livestock industry, it's not as publicized and studied as it should. Yeah.
Interesting.
And that's the other reason why mink farmers now are very wary of people studying SARS-CoV-2 there as well.
I see. Makes sense.
So, do you feel like this is Bill Nate, do you feel like there is a major worldwide decline in immune function? And if so, what are the
Decline
markers of that?
in immune function in mink? Is that what you mean, Bill?
No, in anyone.
I don't think it's a immune decline. Like mink and ferrets, as you know, have been animal models for the study of influenza and other viruses because they have respiratory receptors that are quite similar to those in humans. And so it's just the chance that we also happen to farm these animals in really high densities. And if they are susceptible to the viruses that we have, it's normal to have, these infections. And then that's boom into outbreaks there.
Well, aside from these zoonotic sources, if absolute immunity is behind the rise in variants,
I don't think that question is. I don't think that no one knows that. There's not really shown a big decline in immunity globally. No one's just saying that. It's just saying these viruses can be transmitted, as Nadia said, sometimes easier, between humans and mice.
Yeah,
I mean,
I don't
humans
think there are many studies on that, as you said, Afshin, but I know there are studies that are just starting looking at seroprevalence and exposure of a variety of populations, human and animal, to certain pathogens. But it's different. It's on the other side, yeah.
And that's something that's also a topic that people, like you said, right? Mm-hmm. Overexposure to that population could cause, you'd be potentially more susceptible, right, over time. Like people developing allergies to mice who do a lot of mice studies, for example, right?
Yeah.
That's a whole different thing. That's not viruses, that's just allergies. And Sunny had asked, Sunny, why don't you ask your question in the chat, because maybe someone here might have an answer. Yeah, yes, I don't think there was. Go ahead.
Thank you. Yes, going back to our conversation on environment effects on SARS-CoV-2 in different ways, mutation into different variants and spread. Have there been any correlation studies exploring, for example, SARS-CoV-2 spreading depending on location, say in a tropical environment like with India and Brazil was mentioned, versus in an environment that's more like a desert, like Australia is on average? Have there been any kind of correlation studies in looking at those different factors?
I can try to answer that. So not very formal studies looking like have been done for flu, but there have been some association of climate and SARS-CoV-2 waves, and that is just due to the observable data where we have bigger pandemics, at least in the northern hemisphere, in winter versus in the summer season. But I see that Chris just sent something, so maybe there's more info there.
Chris just put an article here that I think we went over this a while back too. Some papers who came out that talked about humidity and how well does humidity transmit the virus.
Right.
There's a few papers, and this is a nice one, I think. Yeah, I saw those before the Chris things.
Yeah,
So
indoor study.
it does have the. Yeah, sorry. Go ahead, Nadia.
No, I was just acknowledging Chris' comment. This is an indoor study, and yes, for sure there are quite a few of those, but not like at a global scale. Try to relate to climate patterns.
But as Chris says in the chat, it could give some clues, maybe. There's some feedback here. If someone can mute their mic. Oh, yeah, sorry. All right. Yeah. So I guess. Gotta mute the mic here. Much feedback. So yeah, and then Sazwadi also pointed out in India the 10,000 new cases today, and it's majorly XBB.1.16, going back to the original article. So yeah, I guess you're in the fourth wave there. And also Sazwadi, you put another link and article in the chat people could look at, talking about the environmental correlations too. So. Oh, this one's talking about pan-continents, so maybe this might address some of Sunny's questions there. Thanks for that. All right, so let's keep going. Good discussions. Feel free to interrupt that more. The wastewater in the U.S. is still showing it down. And once in a while, I see these little blips that go up. But it's, again, the good news is in the U.S. at least, it's well, we don't have, as you see here, XBB.1.16 is only 4%, 5%. But right now, it's showing the wastewater, the steady decline, as we've been seeing the past, since January, keeps going down. So that's good. One of the counties where I live in, for example, you see these little blips that go up and down, but it keeps going down, which is promising. But that seems to be a trend all over the U.S. So that's some good news. Let's see. If XBB.1.16 comes over in the US, what happens to this? But for now, good news, this is just a breakdown of the variance. As we saw in that previous plot, again, this is XBB.1.5, you see .1.9.1. I guess the CDC classifier, I think, has not yet classified 1.16. I'm trying to read the footnotes here, but I don't think they have from last I heard, so that's why it's not showing up here, I think. The trends globally, this is just looks like it's all decline, but if you go more of a macro scale, you see, I mean, again, the case numbers are not as reliable these days, I think, everywhere. So nowhere's going to show actually a spike in cases reporting, places like our world in data.org and other places, they're not going to show that accurately because of the lack of reporting. But if you look at hospitalizations, you see these are in decline, which is good. Like in France, for example, you see hospitalizations are slowly going up, but this could be a little blip there. Maybe this is because of new variants coming in. I don't know. Germany still has decline. US, we've already been over decline. Brazil and South American countries usually follow the same trends. Decline there, but of course they don't report their hospital admissions or ICU, so we don't have this data. Japan has their decline. I think Japan still reports cases fairly accurately, but I could be wrong. But the ICUs are for patients, patients in ICU are down too, so that's good. And Singapore had their spike. This is their spike for 1.9.1, XBB.1.9.1. This last little bit going down, I don't think is this noise from the data coming in, but this is the spike that because Singapore still is doing pretty well as reporting cases, but you can see the spike happening because of XBB.1.9.1. India I don't show because they just are not having a very good reporting of it. But all the reports, as Wadi said in the chat, 10,000 cases today. From last week to this week, there's like 40,000 new cases in India due to XBB.1.16. That's it. And unless people have any other comments and questions, now you can let me know.